Alright, so we are back. Now I'm going to talk about the different types of biological waste and how to handle them. Okay, so in a lab we have lots of waste. We have sharp waste, we have non-sharp waste, we have liquid waste, solid waste, and they fall in different categories. Okay, I'm going to go through one by one. Okay. Alright, the first category is the sharp waste. Okay, sharp instruments such as syringes, needles, scalpel, and other sharp instruments, all that can cause injuries, they fall into category A. Okay, category B, they are the non-sharp waste, such as gloves, tissue papers, petri dishes, plastic culture containers, and hardened agar. Agar is the jelly inside those petri dish. Um, so when they are hardened, they are considered non-sharp waste. Okay. Next one is category C, animal carcasses. So we have, if we decide to dissect the frog this year, or the rat or the fish, all those animal carcasses falls into category C. And the last category is liquids. So broth culture, broth, I mean in Bahasa we call it kuah, but broth culture means it's a food mixed liquid. And then liquid mediums such as blood is also considered as liquids. Okay, so these ways, different categories, there are different ways to dispose of them. Now for category A, okay, they are placed into a special bin for sharp material disposal. This bin does not need to be sterilized. Okay, remember that. Sterilized means you don't have to disinfect them. Okay, you don't have to kill all the bacteria or anything. Just put it into the sharp waste bin and they will be incinerated. They will be destroyed. Okay, for category B, they are packed first in an autoclave resistant biohazard plastic bags. Okay, sterilized in an autoclave for decontamination and then placed into a biohazard bin. Okay, biohazard plastic bags cannot be thrown into regular waste baskets. I'm going to show you the pictures right here. Okay, they are packed first into a what do you call it? Autoclave resistant biohazard plastic bags. This one right here. Okay, and then they are sterilized in an autoclave. So this is an autoclave. It looks like a washing machine, but what it does is those things inside the biohazard plastic bag, okay, they are put in there and they are steamed to a very high temperature, and that way it can kill all the bacteria and germs inside those bags. Okay, so autoclaving is done at a temperature of 110 degrees Celsius and pressurized at 15 psi for 20 minutes. Biohazard plastic bags that have been sterilized in an autoclave and sharp waste bin must be stored temporarily in a controlled storage place until the scheduled time for disposal. Okay, so basically autoclave, it's like pasteurization for milk, but this one is for waste. Alright, for category C, animal carcasses, they are wrapped carefully in an absorbent material such as tissue paper. They are packed carefully into a biohazard plastic bag and frozen. Okay, so now you know why the movie is called Frozen. Okay, just kidding. Alright, then we have category D, liquids. They are also decontaminated by autoclaving before disposal. And then the sterilized liquid waste must be disposed immediately. Okay, so category B and D needs to be autoclave. Right? C for animal carcasses, they need to be frozen. And for category A, sharp waste, you throw it in a metal plastic bag. Okay. All right, let's proceed to the next one. Accidents in the laboratory. I'm not sure if we need to go through all these things. That some of the very important thing that you have to know is when there is a mercury spill, please inform your teacher. Okay, declare the spill area as a restricted zone. Sprinkle sulfur. Okay, to cover the mercury spills if you have access to sulfur. Okay, and then call the fire and rescue department. So yeah, that's this is very very important. Mercury is not it's not a plaything. Okay, clothing ethics, blah blah. Laboratory safety rule. I think this is fairly straightforward. Most of them do not require, I won't require you to do all of these things because we don't usually do them, but please do not drink and eat in the laboratory. Maybe sweets can, I guess, chewing gum. Identify all safety symbols on substances and equipment before use. Okay, fires, don't panic and stay calm. Okay, do not play with the emergency switch. Okay, like the one we had last time. Handling glass and chemicals, be cautious. Handling live specimens, use appropriate gloves. All surfaces and workstations should be cleaned with disinfectant. Okay, very important. You don't want to have germs all over the place after you dissect a mouse or a frog, for example. Okay. Emergency help. Just inform your teacher, and I'll take care of the rest. Okay. Remove the victim from the scene. If the victim is close to fire, you want to remove the victim from the fire. Emergency treatment. If you are a certified rescuer with BBSM, then you do that. If don't, then don't touch anything. Okay. Scientific attitudes and noble values. Now, some teachers probably just let go of this thing, but for me personally, I think these are the most important part of being in a science stream, being a science stream student. Okay, number one, you must have interest and curiosity towards the environment. Okay, what makes you a good scientist is having all these how many? 1, 2, 3, 4, 5, 6, 7. Okay, 7 attributes. Okay, 3 of this, okay, you've learned in Form 1. Okay, number 1 is having interest and curiosity. Number 2, being honest and accurate and recording and validating data. Okay, now sometimes it's very how should I word this thing? You're very tempted to falsify your data, okay, because this data does not look correct. Okay, I should tweak this data a little bit, change the numbers a little bit. Don't do that. Okay, a good scientist is always honest in recording their data. Even though it's wrong, it does not prove their hypothesis. Okay, maybe they did the procedures wrongly. That's why it's very important on being honest. Okay, sometimes you don't catch those mistakes if you do everything correct. Okay, number 3, being diligent and persevering in carrying out a study. Diligent, what does diligent means? I'll get back on you with that. Number 4, being responsible. If you break a thermometer, please be responsible. Don't blame others. Okay, appreciating and practicing clean and healthy living. Of course, appreciate the contributions of science and technology, and think critically and analytically. Okay, these 4 these 3, I think it's very straightforward, but the first 4, not the first 4, this one, and this one is very very important, in making you a good scientist. Now, a good scientist does not necessarily mean that, I need to be in a lab to be a good scientist. No, a good scientist is practicing good things towards science. Okay, all right. Okay, 1.3, I'll do it in the next video.